Another class of cancer drugs may contribute to curing HIV. Two drugs from a class new to HIV medicine called BH3 mimetics were unveiled at July's 12th International AIDS Society Conference on HIV Science, IAS 2023, in Brisbane. They may contribute to a cure for HIV by killing off long-lived cells that contain HIV genes in their DNA. Notably, venetoclax, Vinclixta, and obadoclax only killed off cells containing intact DNA, capable of giving rise to new viruses, and did not delete cells containing defective, harmless DNA. A number of drugs and treatments from the anti-cancer arsenal have been investigated as HIV cure research, such as TAC inhibitors, PD-1 inhibitors and therapeutic vaccines. And, of course, the 6 successful cures so far have used the radical cancer therapy of a stem cell, bone marrow, transplant. This is not coincidental. Cancer and AIDS are both the end result of mutations in the DNA of some of our cells. In the case of cancer, they arise in the host DNA, and in HIV infection they are introduced by a virus, but both are the result of rogue genes. Some other viruses, such as HPV, directly cause cancers. Cures for cancer and HIV share one other property. They are difficult because the immune system finds it harder to see cancer and HIV-infected reservoir cells, as they do not show most of the obvious distress signals that virally infected cells usually do. Drugs that specifically target HIV-infected cells would take us a long way towards a cure. In the case of the BH3 mimetics, HIV cure researchers noticed a similarity between cancer and HIV reservoir cells in that both seem to be immortalized. Something seems to stop them undergoing the normal and necessary pruning of cells called apoptosis, which kills off cells that have outrun their usefulness or whose senescence may cause malfunctions. It was already known that the immortality of some cancer cells depends on proteins belonging to a family called BCL2, which switch off the process of apoptosis. BCL2 proteins are necessary for growth and healing when needed, but are hugely overabundant in cancer cells. They were first spotted in B-cell lymphoma, hence the name. Normally the activity of BCL2 proteins is regulated by members of the same family called Bax proteins that act as their anti-selves, slotting into a cleft in the BCL2 and blocking the protein's anti-apoptotic action. Cancer researchers discovered a class of small molecules called BH3 mimetics that slot into BCL2 proteins in the same place as Bax. It's called the BCL2 hydrophobic pocket number 3, hence BH3. Hypothesizing that HIV-infected cells become long-lived reservoir cells that evade immune surveillance due to the action of BCL2, researchers have wanted to see if BH3 mimetics could disrupt that process. Venetoclax. Venetoclax is furthest along in the research pathway because it is already a licensed treatment for certain types of leukemia under the brand names Vinclixta and Vinclixto, and has thus cleared safety hurdles. Dr. Philip Rangelovic of Australia's Walter and Eliza Hall Institute of Medical Research presented the results of a study of venetoclax in 14 mice humanized so their immune systems were receptive to HIV infection. The mice were infected with HIV, then placed on 3-class antiretroviral therapy, tenofovir/imtricitabine/rilpivirine/raltegravir. As it would with humans, it took about a month for them to become fully virally suppressed. While still on ART, 8 of them were given venetoclax and 6 of them a placebo, over 7 weeks. These were given on a 5 days on, 2 days off basis, and there was also a pause of a week in the middle. This was to avoid intolerance as far as possible. Cytotoxic drugs like BH3 mimetics almost inevitably have some side effects, including a general reduction in all cellular blood components, pancytopenia. After the last venetoclax dose, the mice were then taken off ART. HIV viral loads in humanized mice normally rebound very fast. They have fewer cells to reinfect, and all the placebo animals had detectable viral loads within a week. Rebound was slower in the mice receiving venetoclax. One rebounded after a week, 5 after 2 weeks and 2 after 3 weeks. The same study was repeated with venetoclax and another drug called S63145, an inhibitor of a different apoptosis suppressor called MCL1, found in another cancer, myeloid cell leukemia. The extra drug did nothing in itself, but when the two drugs were combined and given to 4 mice, 2 rebounded virologically after 2 weeks but the other 2 not till 4 weeks. So the median time to rebound was 2 weeks on one drug but 3 weeks on both. In ex vivo tests, DNA-positive CD4 cells were donated by 10 people with HIV and cultured in a lab dish along with 3 different doses of venetoclax, 5, 10 or 100 nanomils, or with the inactive substance dimethyl sulfoxide, DMSO. DNA assays measured the amount of total HIV DNA, intact DNA capable of forming new viruses, and defective DNA. At the highest venetoclax dose, the total amount of DNA declined, relative to cells treated with DMSO, by 61% and the amount of intact DNA by 42%. The former result was statistically significant, p equals 0.031, the second result not quite so, p equals 0.064. Where 0.05 is the conventional upper limit of statistical probability, or less than 1 chance in 20. This was a random finding. In contrast, there was no decline at all in the amount of defective DNA in the cells. This last finding is the most interesting one as it suggests venetoclax is doing a lot more than indiscriminately destroying cellular viral DNA. It suggests that by selectively inhibiting BCL2 in cells that contain too much of it, it also selectively marks out only those immune cells that contain viable HIV DNA and ignores cells containing only fragments of it. This ability to target active or potentially target active reservoir cells is one of the most essential requirements of the most feasible and scalable HIV cures.